chemistry of macromolecules. Cellular macromolecules are large molecules that are essential for life. They are the building blocks of cells and therefore the building blocks of everything that you are. We'll spend some time looking at their chemical structure because physical and chemical properties inherent to molecules influence the types of interactions possible with other structures within individual cells or between organisms. The chemical structure of molecules determines their function. The four major macromolecules are carbohydrates, proteins, lipids, and nucleic acids. You see images of the chemical structure on this slide. One of the things you should notice right away is that they're all organic, i.e. carbon-based. So organic in this context means something very different from like organic food in the grocery store. Macromolecules are formed by combining small units, also called monomers, into large molecules called polymers. Mono meaning one, poly meaning many. Dimers and oligomers are another term that you'll hear a lot. Di means two, oligo means a few. This chemical process that combines them together is called dehydration synthesis. Dehydration synthesis releases water, and when the polymers are broken down, the water is being added back. The interaction of these molecules with water is extremely important. Some macromolecules easily interact with water. We call those hydrophilic. Others repel water. We call those hydrophobic. And still others have portions that are hydrophilic, while other portions are hydrophobic. Lipids as a group are hydrophobic. You should know what monomers, or the chemical building blocks, are that make up these polymers. So carbohydrates are made from monosaccharides. Proteins are made from amino acids. Lipids are made from hydrocarbons. And nucleic acids are made from nucleotides. You know many of these molecules already from the nutrition labels on the foods that you eat. We get energy from many of these molecules when we eat them. The energy contained in the molecules is actually contained in the carbon-hydrogen bond. Looking at the chemical structures on the right, can you imagine which of these molecules are probably the most energy dense? Looking at a nutrition label, one of the things I've got to tell you is that energy comes from, when it comes from food, different energy is included in the different molecules. So fat has 9 calories per gram, carbohydrates 4 calories per gram, and protein 4 calories per gram. And when you're looking at the chemical structure, note that fat has way more CH bonds than carbohydrates or protein have. So this is why fat is the most energy dense. But we'll start with carbohydrates. Carbohydrates are found in fruits and in starchy vegetables and grains. The biological function of carbohydrates are energy storage, energy production, cell signaling, and structure. And let's go look at the chemical structure of carbohydrates. Carbohydrates, as the name implies, contain carbon and water. They also contain oxygen. The ratio between carbon to hydrogen to oxygen is 1 to 2 to 1. Most carbohydrates exist as ring structures in biological solutions, and we can classify them into 3 subtypes. Monosaccharides, like glucose, for example, disaccharides, like sucrose, and then polysaccharides, like starch, glycogen, and cellulose. Whether you realize it or not, glucose is the most important sugar in your life. It's a universally used sugar, and it is what all other sugars are converted into to make ATP, which is the energy currency in your body. Glucose can freely donate electrons and is referred to as a reducing sugar. We'll talk about oxidation reduction in just a second. Sucrose is a disaccharide made from glucose and fructose. And it's made through the process of dehydration synthesis that I talked about earlier. In the figure on the right, you see how the removal of water will combine the two molecules into a disaccharide. Sucrose is found in fruits, sugarcane, beet, and it is what is our table sugar. So the sugar that you use at home, that's table sugar, that's sucrose. Other well-known disaccharides are lactose, which is galactose plus glucose, and you find that in milk. And then maltose, which is found in grains, which is a glucose-glucose disaccharide. The polysaccharides you probably heard of, also called complex carbohydrates, are starch, which plants use for storage, glycogen, which animals use for storage, and cellulose, which plants use for structure. All three complex carbohydrates are made from glucose molecules, but the molecules are attached to each other in a slightly different way. The main difference between starch and glycogen is the level of branching. In cellulose, the glucose molecules attach to each other in a different way. And while we can digest glycogen and starch, we cannot digest cellulose because we don't have the enzyme tools to break that molecule apart. You can think of cellulose as the fiber in your food, so it's the roughage, it's the stuff we can't digest. By now, I'm pretty sure you're pretty sure that you've accidentally signed up for a chemistry class, but we have a couple of reactions that we're using to detect macromolecules that utilize an oxidation reaction, and I think it may be useful to go over oxidation reduction for just a moment. The terms oxidation and reduction are actually quite unfortunate. And that's why most people just remember the mnemonic. So if nothing else, remember the mnemonic. Oil rig. Oxidation is the loss of electrons. Reduction is the gain of electrons. Oil rig. But let's run through what's actually going on. The reason why the gain of electrons is called reduction actually has historical reasons. When alchemists and medieval metallurgists started doing experiments to quantify exactly how much iron was formed from smelting a given amount of iron ore, they found that the weight of the resulting iron was always less than that of the ore, ergo reduction. When we later learned about electrons, the term remained even after we realized that the iron was actually gaining electrons even though it lost weight. When I think about the process of oxidation reduction, I look at what oxygen does. When you look at oxygen in the periodic tables right there, you see that it has 6 valence electrons in its outer shell. It wants 8 and to fill the shell. And oxygen will do just about anything to grab 2 electrons from someplace else. The fact that it wants that is called electronegativity. So oxygen is very, very electronegative. And that electronegativity, by the way, is also the reason why water is polar. So oxygen oxidizes things by stealing electrons from other atoms. The atom that lost its electron is said to be oxidized, while oxygen is said to be reduced. So if you want to think about this mathematically, you can try to remember that gaining electrons makes something more negative because electrons are negatively charged. Or just remember oil rig. Now that that's clear as mud, let's look at copper for just a moment. The Statue of Liberty is made from steel that was covered with copper. It used to be red, like a shiny penny, but over time it became green. That's due to oxidation. It turns out that copper experiences color change when it is oxidized. It goes from red to green when it's oxidized and from green to red when it's reduced. Reduced meaning it gains electrons. And in our test, we're going to take advantage of one of those color changes. But before we go to that, let's talk about reducing sugars real quick. Reducing sugars do the opposite of what oxygen does. They happily give up electrons. So reducing sugars are easily oxidized while the other agent is reduced. Ergo, reducing sugars. Not all sugars are reducing sugars. In order for a reducing sugar to be able to give up an electron, it has to be in a hemiacetal form. Hemiacetal means that you've got an OH group attached to the same carbon as the OR group, as opposed to two OR groups. R stands for rest here in this context. Glucose is a reducing sugar because it has that hemiacetal. Because it's a reducing sugar, it exists in a ring form as well as in a linear form, and it kind of freely goes between those two forms in solutions. All monosaccharides are reducing sugars, and some disaccharides are. Sucrose is not. Sucrose has an acetal form here, so it's got two OR groups attached to this to the carbon. So it's in a locked position, and it can't do anything. Our test will allow you to distinguish between glucose and sucrose, and interestingly enough, it will also allow you to distinguish between sucrose and high fructose corn syrup, because high fructose corn syrup consists mostly of glucose and fructose, which are monosaccharides, which are reducing, and sucrose is not. So Benedict's reagent is our reagent that tests for reducing sugars. Benedict's reagent is slightly blue, and that's because it contains copper 2+ ions. And this copper 2+ is being reduced to copper 1+, and then to copper without any pluses. And so the copper, as more and more reducing sugars are in the solution, it's actually semi-quantitative. So a little bit of reducing sugar will turn it green. Orange means it's a little more, and when it's brick red, that means there's a large amount of reducing sugar there. So you would expect a color change with glucose, but you would not expect a color change with sucrose. And would you expect a color change with high fructose corn syrup? You can answer that question yourself. The next test is the test for starch. Starch is a complex carbohydrate that's slightly branched, consisting of amylose and amylopectin. Iodine forms a starch-iodine complex with amylose, and that causes a black color to develop. So the black is the positive. Over on the right, you see an apple slice, that's your negative control, and a potato slice, that's your positive control. So iodine, apples does not contain any starch. Apple contains mostly glucose and fructose. And so the iodine does not have a color change with the apple, but it very much has a color change with the potato. The next macromolecule we're going to look at is protein. You'll find protein all over your body. They're part of your flesh, your skin, your hair, your nails, your teeth. The animals we eat are basically made out of protein. Leather is made out of protein, so is wool, silk, and spiderwebs. Insulin is made from protein, so are the clot-busting enzymes given to patients after heart attacks. Proteins are used in industry to produce high fructose corn syrup or fuel-grade ethanol. So proteins are everywhere, and they can do all kinds of different things. When you look at proteins, the chemistry of proteins, they're made from long chains of amino acids. There are 20 different amino acids in our body. We produce 12 of them. That means 8 of them have to come from food. And we call those essential amino acids. Anytime you need to get something from food, whether that's an essential vitamin, an essential amino acid, essential means you can't produce it yourself. When you look at an amino acid, they all have the same basic structure that's shown here off to the right. So you've got the carbon in the middle, and then you've got an amino group, and you've got a carboxy group. And then there's a rest. And the rest determines the specific characteristics of the amino acid. amino acids are attached to each other by dehydration synthesis again. And the bond they form has a specific name. It's called a peptide bond. Hundreds of amino acids, sometimes thousands of amino acids together make up proteins. And the proteins form three-dimensional structures. There's an example of some of these three-dimensional structures on the top left. And on the top right is actually a electron microscopy picture of a hemoglobin molecule. So this is what carries oxygen in your blood. The test we're doing for protein actually tests for the presence of the peptide bond. So the test is called Biuret. And Biuret contains copper sulfate, which provides the copper 2 ions needed for this reaction, potassium hydroxide for alkalinity, and potassium sodium tartrate for stabilization. And what happens is that the copper 2 ions, they form a chelate, a chelation process complex with the peptide bond. And that absorbs its 540 nanometers, which happens to be purple. So that's what makes this solution purple. Now you need a minimum of 3 amino acids to actually make this peptide bond work and to make this complex work. So the if the protein chains are too short, this sometimes actually can be pink, but in our case, we won't see any pink. Chemically, lipids are a little different from the other macromolecules. They are a family of compounds that just don't dissolve in water. They possess many CH bonds, which means that they're very high energy rich. We store energy in lipids in our body and as triglycerides. They're also important for insulation for the body. So this walrus here on the right-hand side, for example, has a lot of fat because it keeps it insulated against the environment. We have fats as cushions around bones and organs. They're also important for fat-soluble vitamins. We use lipids commercially in the production of soap and cleaning products. Our steroid hormones are based on lipids. And phospholipids are crucially important for every cell in your body. So lipids are a family of compounds that don't dissolve in water. They're very energy rich. Fatty acids, when you look at a fatty acids, it has 2 parts to it. It's got a carboxyl group, which is hydrophilic, which is this part here, which is slightly polar, which is what makes it hydrophilic. And then it's got the methylene group, which is hydrophobic. Fatty acids can be saturated or unsaturated. Saturated fatty acids tend to be straight. Unsaturated fatty acids tend to be bent. And because you can pack straight things closer together than you can bend things, you find saturated fatty acids mostly in things like butter, lard, the solid fats, whereas you find unsaturated fatty acid mostly in the oils, like your olive oil or, that kind of thing. 2 types of lipids I want to specifically look at are triglycerides and phospholipids. Triglycerides are used for energy storage. A triglyceride is a glycerol that has 3 fatty acids attached to it. And then phospholipids. Phospholipids really is the molecule that makes the world go round. Phospholipids are very similar to triglyceride in that they all have a glycerol group, but they only have 2 fatty acids attached. And the third thing that's attached to that glycerol group is a phosphate group. When you look at the structure of this thing, what you end up with is this hydrophilic head and these hydrophobic tails. And when you take phospholipids and you just stick them in a soup, what they do is all the hydrophilic parts go together and all the hydrophobic parts go together. And so they naturally form this thing that we call a micelle. And this micelle, if you know anything about cells, right, you can immediately tell that this is basically a very early version of a cell. This has what you need. You need an outside and you need an inside, because you can have reactions going on in the inside that don't go in the outside. The test we use for lipids is Sudan 4. Sudan 4 is a lipid-soluble red dye. It does not dissolve in water. And so if you learn anything at all from this class, is that like things dissolves in like things. So Sudan 4 dissolves in things that are lipid. So if you have a lipid, Sudan 4 will dissolve in it and you'll have a red color throughout. If you have water or a water-based solution, Sudan 4 will not dissolve in it, and so you end up with 2 layers. The last molecule we're going to be talking about are nucleic acids. Those are found inside of the cell. Those are the genetic material. Our genetic material is DNA. Some organisms' genetic material is RNA. The genetic material is made out of nucleotides, which are base paired on a sugar phosphate backbone. Now, the test we're doing for nucleic acid is actually specifically a test for the presence of GMO. And the way glyphosate works is normally when you have a plant, they need a way to make the amino acids phenylalanine, tyrosine, and tryptophan. And what the glyphosate, what the Roundup does is it interferes with the activity of this enzyme that's involved in this process. Now, the herbicide-resistant GMO plant has a replacement enzyme here. It's a modified enzyme that's called CP4-EPSP. And what we're doing in the test is we're testing for the presence of this protein, this modified enzyme. The test is an immunochromatographic test strip. And what you basically do is you have a sample, and then you wick the solution up this way. The protein, the GMO protein, the CP4 protein, if it's in the extract, it will bind to an antibody that has a colored particle attached to it, and because antibodies are very, very specific. And so if it exists, then there's an antibody particle conjugate that happens, and then that conjugate is being caught at a capture location and forms the CP4 band if it exists. There's a second band there that is a control band that just shows you that this thing is working. This is a very similar test to the way a pregnancy test works. So a pregnancy test, the 2 bands, one of the bands just tells you the test is working, and the second band tells you whether you're pregnant or not.